The human body uses carbohydrate, fat and protein in food and from the body stores for energy to full physical activity. These essential nutrients are needed regardless of the intensity of the activity you are doing. If you are laying down and reading a book or running a marathon, these macronutrients are always needed in the body. However, in order for these nutrients to be used as fuel for the body, their energy must be transferred into the high energy molecule known as adenosine triphosphate, which is ATP. ATP is the body's immediate fuel source and can be generated either with aerobic metabolism in the presence of oxygen or anaerobic metabolism without the presence of oxygen. The type of metabolism that is predominantly used during physical activity is determined by the availability of oxygen and how much carbohydrate, fat and protein are used. Carbs metabolism during exercise. Carbohydrate metabolism is biochemical process that makes sure living cells have a constant supply of energy. Next, the breakdown of carbohydrate will make a glucose and it is the essential fuel for energy production in the contracting skeletal muscle. Glucose can be broken down by glycolysis and used to generate ATP by the Krebs cycle and oxidative phosphorylation. The breakdown process of glycogen is glycogenolysis. For the information, liver and muscle store the highest glycogen percentage in the body, which is 400 gram and 100 gram. Function carbs metabolism during exercising. First one is main storage form of glucose in human body. Source of energy for human body. Sparing the use of protein in human body. External protection for heart and good health. And last one is part of the nucleic acid building blocks. Test of carbs metabolism during exercising is carbohydrates intakes and intensity and duration of exercise. Breakdown process carbs metabolism is glycogenolysis. Breakdown process of glycogen to glucose 1. Phosphate and glucogen N1. Supply energy directly to the skeletal muscle. Regulate hormonally in response to blood sugar level by glucogen. Stimulate by epinephrine during the fight or flight response while exercising. Exercise has an impact on carbohydrate metabolism because of the increased demand for energy supply to the skeletal muscle. The rate of glycogenolysis will increase. Because of the high rate of consumption during exercise, the amount of glycogen in the body will increase. Athletes should consume carbohydrate after exercise to maintain the body's stored quantity of carbohydrate metabolism. For the conclusion, the rate of carbohydrate synthesis increase during exercising to make sure the constant supply of energy to the skeletal muscle. Next, we go to the protein. Protein are responsible for nearly all bodily and cellular function from structure proteins in bones, contractile proteins in muscle, transport protein in blood plasma to hormones, antibodies, cell receptors, ion channel and enzyme that catalyze almost every chemical in biological system.
Proteins are polymers of amino acids linked together by peptide bonds. An amino acid consists of an amino group, a carboxyl group, and a unique side chain connected to a central carbon, the alpha carbon. Instead of being an extended chain of amino acids, a protein usually folds into a three-dimensional conformation that is critical for its functions. The structure forms as a result of interactions between the side chains of amino acids, and is thus dictated by the amino acid sequence.
Metabolic state before starts has many factors, which is dietary composition, when your last meal, circulating insulin and glucagon, other hormones, recent exercise, body composition, and basal metabolic rate. What changed when you start? Increase in sympathetic activity, which is epinephrine or adrenaline, fight or flight, increased heart rate, cardiac output, respiratory rate, bronchodilation, shunt blood to skeletal muscle, mobilize glycogen, some fat breakdown, protein metabolism, which is synthesis, surprise, degradation, increase. Exercise has a profound acute effect on protein metabolism. Leucine oxidation is increased during exercise, thus indicating exercise net protein breakdown. Following endurance exercise, whole body protein breakdown is generally reduced from resting level, while following eccentric exercise, both whole body protein breakdown and leucine oxidation are increased. Effect protein metabolism after exercise. Catabolic activity becomes slow, which is decreased glucose demand. Net protein breakdown slows, protein synthesis increase. This is when hypertrophy occurs. Make sure to maintain high dietary protein and amino acid intake. Conclusion: generally protein synthesis suppressed during exercise. Protein breakdown is increased to support glucose synthesis and modified by many factors, including hormonal activity. Next, we go to the lipids metabolism. What is lipid metabolism? Entails the oxidation of fatty acid to either generate energy or synthesize new lipid from smaller constituent molecules. Associated with carbohydrate metabolism, as a product of glucose can be converted into lipid.
Although the term lipid includes several types of molecules, lipid metabolism usually refers to the breakdown and synthesis of fats. Fats are triglycerides. They are esters of glycerol and 3 fatty acids. Fats can come from the diet, from stores in adipose tissue, or can be synthesized from excess dietary carbohydrates in the liver. Dietary fats are digested mainly in the small intestine by the action of bile salts and pancreatic lipase. Bile salts emulsify fats. They act as a detergent, breaking large globules of fat into smaller micelles, making them more accessible to lipase. Pancreatic lipase then converts triglycerides into monoglycerides, free fatty acids, and glycerol. These products move into the cells of intestinal epithelium, the enterocytes, inside which they recombine again to form triglycerides.
Endogenous triacylglycerol is represent an important source of fuel for endurance exercise. Triacylglycerol oxidation increase progressively during exercise. The specific rate is determined by energy requirements of working muscle, fatty acid delivery to muscle mitochondria, and the oxidation of other substrates. The catecholamine response to exercise increased lipolysis of adipose tissue triacylglycerol and presumably intramuscular triacylglycerol. Increase in adipose tissue and muscle blood flow decrease fatty acid restriction and facilitate the delivery of released fatty acid to skeletal muscle. Alteration in fatty acid mobilization and the relative use of adipose and intramuscular triacylglycerol during exercise depend in large part on degree of fitness and exercise intensity. Compare with untrained persons exercising at the same absolute intensity, persons who have undergone endurance training have a greater fat oxidation during exercise without increased lipolysis. Available evidence suggests that the training-induced increase in fat oxidation is due primarily to increased oxidation of non-plasma derived fatty acids, perhaps from intramuscular triacylglycerol store. Fat oxidation is lower in high-intensity exercise than in moderate-intensity exercise, in part because of decreased fatty acid delivery to exercising muscle. Parenteral lipid supplementation during high-intensity exercise increase fat oxidation, but the effect of ingesting long-chain or medium-chain triglycerol on substrate metabolism during exercise is less clear. Fat is an extremely important substrate for muscle contraction. Triglycerides stored in adipose tissue and within muscle fiber are considered to be the main source of the free fatty acid oxidized during exercise. The regulation seems to be multifactorial and includes dietary and nutritional status, hormonal milieu, exercise mode, intensity and duration, and training status.
Metabolism pathways are closely connected to those of carbohydrate metabolism. Glycerol is converted to a glycolysis intermediate, while fatty acids undergo beta oxidation to generate acetyl-CoA. Each round of beta oxidation removes 2 carbons from the fatty acid chain, releasing 1 acetyl-CoA, which can then be oxidized in the citric acid cycle. Beta oxidation also produces several high-energy molecules, which are fed directly to the electron transport system. Fats yield more energy per unit mass than carbohydrates. When acetyl-CoA is produced in excess, it is diverted to create ketone bodies. During glucose starvation, ketone bodies are an important source of fuel, especially for the brain. However, ketone bodies are acidic, and when produced in excess can overwhelm the buffering capacity of blood plasma, resulting in metabolic acidosis, which can lead to coma and death. Ketoacidosis is a serious complication of diabetes, in which cells must oxidize fats for fuel as they cannot utilize glucose. Extreme diets that are excessively low in carbohydrates and high in fat may also result in ketoacidosis. On the other hand, diets that are high in carbohydrates generate excess acetyl-CoA that can be converted into fatty acids. Synthesis of fatty acids from acetyl-CoA is stimulated by citrate, a marker of energy abundance, and inhibited by excess of fatty acids. Fatty acids can be converted into triglycerides for storage or synthesis of other lipids by combining with glycerol derived from a glycolysis intermediate.
For the conclusion, during exercise, carbohydrate is a key energy source. Factors like metabolic state, diet, type, intensity of exercise, and training status all affect its use during exercise. For the lipids and fats are critical components of the human body's homeostatic function. Lipids have a role in many of body's most important functions. Also, same like protein is a very complex molecule found in all living things. Protein have a high nutritional value and play a direct role in a chemical reactions that keep life going. A protein molecule is considerably larger than a sugar or salt molecule, and it is made up so several amino acids link together to create lengthy chains, similar to how beads are strung together. These all 3 components are very function to our body during exercise or do homework in our life.